Good afternoon, students. Last time we discussed about conjugation. What is conjugation? It is a genetic recombination in which there is transfer of DNA from a living donor bacterium to the recipient bacterium through a conjugation tube. Okay, the conjugation tube is formed because of the sex pili, and sex pili can be produced by the bacteria, which is specified by a typical type of plasmid called as F plasmid or F factor. And the cells having the F factor are called as F plus cells. Okay, so there is a physical contact between the donor and the recipient cell, and then only the DNA is transferred from donor to recipient. Okay, now as far as conjugation are concerned, we discussed last time that there are three different type of donor cells: F plus cell, HFR cell and F prime cell. So last time we discussed about F plus cells and their conjugation or their mating or crossing over with F minus cell. Okay, so today we are going to discuss about HFR cell. What happens in when a F plus cell crosses with F minus cell? F minus cell is converted into F plus cell. But recombination does not take place because when F plus cell crosses with F minus cell, the F factor, that is plasmid, is transferred from donor to the recipient. The recipient also becomes F plus, but the chromosomal genes, chromosomal DNA from F plus is not transferred to F minus. So the chances of recombination taking place are very less when F plus cell crosses with F minus cell. Then how do recombination take place by the process of conjugation? There are another type of cells which are also donor cells. They are called as HFR cells, high frequency recombinant cells. Okay, HFR cell, with HFR cell as donor cells, the chances, the frequency of recombination is very high. Okay, so the chances of recombination taking place with HFR cell as donor cell are very high, and hence they are known as high frequency recombinant cells, and the process is called as high frequency recombination. What is HFR cell? Okay, HFR cell is a bacterium in which the F factor, the plasmid, is integrated inside the chromosomal DNA. Okay, what is HFR cell? The cell in which the conjugative plasmid, that is F factor, is integrated inside into the chromosomal DNA. The integration of plasmid into the cell's chromosome takes place through homologous recombination. Okay. Now just consider that this is a bacterial cell. This is the chromosomal DNA. Okay, and this is the F factor or plasmid. Now crossing over takes place and F factor gets integrated inside chromosomal DNA. Okay. So this is chromosomal DNA and the F factor. Okay, so F factor, that is plasmid, is integrated inside chromosomal DNA. Such a cell is called as HFR cell, high frequency recombinant cell. Okay, so you can see here in this diagram in red color there is F factor or plasmid. Such a cell which is having F factor is called as F plus cell, F plus male donor cell. Now in the second figure you can see that there is crossing over taking place, and in the third diagram you can see that the F factor, that is chromosomal DNA, F factor, that is chromosomal DNA, is integrated inside the genomic DNA of the donor cell. Okay, so F factor integrated inside chromosomal DNA, such a cell is called as HFR cell, high frequency recombinant cell. So same here in this diagram you can see that the plasmid gets integrated inside chromosomal DNA. Okay, so this F factor gets integrated inside chromosomal DNA, such a Such a cell is called as HFR cell. Now, how conjugation take place? See, genetic recombination in which fragment of chromosomal DNA from male donor bacterium are transferred to female bacterium following insertion of F plasmid into the nucleoid of donor bacterium. Right? How it takes place? It involves sex pili or conjugation tube. DNA transfer during conjugation is time dependent. Okay. Now let us consider the first point again. What is HFR conjugation? In which fragments of chromosomal DNA from male donor bacterium get transferred into female bacterium, and hence we say that recombination has taken place because the donor cell chromosomal DNA from donor cell is transferred to the recipient cell. The recipient cell receives chromosomal DNA of donor cell, and the chromosomal DNA of donor gets integrated inside the chromosomal DNA of recipient cell, and hence recombination takes place. Okay, donor DNA, donor chromosomal DNA integrated inside recipient cell's chromosomal DNA. So we say recombination take place, and chances of recombination are very high. Okay. Of course, naturally, the DNA transfer takes place through the conjugation tube, which is formed because of F factor. Now, DNA transfer, if a complete strand of DNA of donor is to reach the recipient cell, the time required is very long. Okay, so DNA transfer during conjugation, look at the third point. DNA transfer during conjugation is time dependent. Transfer of entire E. coli donor genome to the recipient requires about 1 hour. But the cells do not remain in conjugation or remain in contact for 1 hour. The conjugation bridge is dissolved before 1 hour, and hence the complete genome or the complete F factor cannot be transferred from donor to recipient, and hence F minus cell remains F minus, but it does not become F plus, but conjugation or recombination takes place. Okay. So transfer begins with an integrated F factor. Okay. And then proceeds in one direction. A part of F factor is the first DNA to be transferred. The part of F factor is followed by chromosomal genes of the donor, and then the remaining F factor. The conjugation tube usually breaks apart long before the entire bacterial chromosome is transferred, and final F is almost never transferred. So the recipient remains F minus. Why it remains F minus? Because the complete F factor is not transferred. What is transferred? First, a part of F factor, then chromosomal DNA of the donor cell, and then the remaining F factor. But before everything is transferred, the conjugation tube breaks. The two cells get separated. So a part of F factor and a part of chromosomal genes from donor go to the recipient cell. Hence F minus cell remains F minus, but recombination does take place. So just look at this. Look at the first figure. You can see in red color F plasmid and the male donor cell. Okay. Now what happens? The F factor, F plasmid gets integrated inside donor bacterium to form HFR cell. So in the first diagram you can see number 1, the F factor integrated inside chromosomal DNA, and hence HFR cell is formed. Okay. In the second diagram, the HFR cell crosses with F minus cell. Okay. Out of the two strands, one of the strand breaks. You can see that the outer strand has broken, and through the conjugation bridge, the outer strand goes to the recipient cell, that is F minus cell. If you look at the diagram, you can see that in the beginning there is a part of F factor followed by chromosomal DNA or donor DNA. Okay, and the remaining F factor is inside, right? So what goes to the recipient? A part of F factor followed by chromosomal genes or genes of donor DNA. Now look at the third. What happens? Through the conjugation bridge, through the sex pili, the conjugation bridge retracts, okay, and two bacteria come closer. One strand of donor DNA goes to the recipient cell, but before the complete one of the strand completely goes to the recipient, the conjugation bridge is dissolved and the donor and the recipient cell get separated. So what gets transferred is part of F factor and part of donor DNA gets transferred to the recipient, as you can see in the fourth diagram. What you can see in the third step? Outer strand is broken. One of the strand of the donor goes to the recipient. What goes? The pink colored F factor followed by violet colored chromosomal DNA. Okay. But before it completely gets transferred, the cells separate. And hence in the fourth figure you can see that donor bacterium has received a part of F factor and part of donor DNA. Okay. The donor bacteria makes a complementary copy of the remaining DNA and it remains HFR cell only. The recipient makes a complementary strand of the transferred donor DNA. Okay. Transferred donor DNA was single stranded. So the recipient cell makes a copy and makes it double stranded. And then what happens? The donor DNA gets integrated inside recipient DNA, and hence we say that the recipient cell has undergone recombination, and that too conjugation. Okay, so the donor DNA fragment undergoes genetic exchange with the recipient DNA, and the donor DNA gets integrated inside recipient cell. Okay, so chances of recombination taking place with HFR cell as donor is very high, and the recipient cell becomes recombinant cell. However, the complete F factor cannot be transferred because the conjugation bridge dissolves, and hence F minus cell remains F minus, but it is a recombinant cell. Okay. Now, you can see again in this diagram, HFR cell you can see. First cell is HFR cell. Okay. In the HFR cell, you can see F factor integrated inside chromosomal DNA. This is F minus cell. HFR cell crosses with F minus cell. Okay, sorry. HFR cell crosses with F minus cell, and the one of the strand from the HFR cell gets integrated or travels through the conjugation bridge and gets integrated inside the recipient cell. Okay. So this is how HFR cell crosses with F minus cell, making conjugation. Okay, through the conjugation tube, one of the strand gets transferred. Okay, but here the chances of recombination are very, very high. The next type of donor cell is F prime cell. What is an F prime cell? See, while the formation of HFR cell takes place, the F factor, that is plasmid, gets integrated inside the chromosomal DNA. Now what happens? The F factor gets separated from the chromosomal DNA. It disintegrates, it excises itself from the chromosomal DNA. As it has entered inside, it wants to come out. So the F factor gets separated from the chromosomal DNA. When the F factor integrated inside chromosomal DNA is getting separated, is getting disintegrated or excised, okay, from the host chromosome, sometimes mistake takes place. Okay, what I am telling you, F factor when gets integrated inside chromosomal DNA of a cell, the cell becomes HFR. Now the integrated F factor wants to disintegrate. It wants to come out of the chromosomal DNA. While coming out, while getting excised, while getting cut off from the chromosomal DNA, mistake takes place. There occurs some kind of a mistake, right? And what happens during this mistake? It is not accurate. Separation is not accurate. What happens? The F factor, the plasmid, by mistake picks up some of the genes of the chromosomal DNA. Okay, so the F factor gets separated from chromosomal DNA, but F factor has carried with it some of the genes of chromosomal DNA. Such a cell is called as F prime cell. Right. So we just discussed that F plasmid is an episome. So once it gets integrated into host chromosomal DNA, it can also undergo disintegration or excise itself back from the host chromosome. Sometimes this excision, this separation, this disintegration is not accurate. It is incorrect. While disintegrating from the host chromosome, the F plasmid might pick up some genes from the host chromosome next to its integration point. So such cells where the plasmid has disintegrated or separated and have got some genes of chromosome is called as F prime cells. They are the derivatives of HFR cell. F prime cells are formed from HFR cell, and F prime cell also serve as donors. So what are F prime cell? Derivatives of HFR cell, where F plasmid has disintegrated from host chromosome, picks up some of the genes of F plasmid, chromosomal DNA. Okay. F prime cells are those in which F factor is formed by excision of F prime factor is formed by excision of F factor from chromosome along with some segments of chromosome. Okay. Now, look at this diagram and see. This first cell is HFR cell, where the plasmid DNA is integrated inside, or F factor is integrated inside chromosomal DNA. In the second diagram, you can see that the F factor is getting separated. The plasmid is getting separated from the chromosome. But there is a mistake in the separation. In the last diagram, you can see that the F factor is having some genes from chromosomal DNA shown by blue color. Okay. The F factor, okay, has some genes from chromosomal DNA that is shown by blue color. Such a cell in which the F factor is having some genes from chromosomal genes is called as F prime cell. Such a cell is called as F prime cell. It is derivative of HFR cell, and it also can serve as a donor cell. Okay. Now what happens when the and as usual, F prime cell also crosses with F minus cell. F prime F minus mating or crossing is similar to F plus F minus crossing. Okay. So here you can see in this diagram that F prime cell is mating with F minus cell. The F minus cell is not having any plasmid. F prime cell is having a plasmid, that is F factor, but the F factor, it is not F factor now, it is F prime factor, because the plasmid is having few chromosomal genes. Now when F prime cell crosses with F minus cell, the F factor gets transferred. Okay. So along with the F factor, what happens? The few genes from the chromosome of donor cell also get transferred. Okay. Few genes from the chromosome of donor DNA also get transferred. You can see here. Okay. The few genes from chromosomal DNA also get transferred. And now the two cells separate. Okay. So F minus cell becomes F prime cell. F minus cell becomes F prime cell. It does not become F plus cell because complete F factor is not transferred. Okay. So F prime and F minus cross is similar to F plus F minus cell, but in this case, along with the F factor, because the F factor here is having few chromosomal genes, the chromosomal genes also get transferred to the F minus cell. Such cells which are having its own DNA. Here the recipient cell is having its own DNA plus some of the genes some of the chromosomal genes of the donor DNA. Such cells are called as partially diploid merozygotes or merodiploid cells. They are called as partially diploid merozygotes. Why? Because they are having, the recipient cells are having their own genetic material plus some of the genes of donor cell. So it is called as partially diploid merozygote. So, and this type of phenomena in which donor DNA is transferred to the recipient cell, okay, as a part of sexual conjugation is called as sexduction. Now you can see here, I'll show you in the diagram. See, this is chromosomal DNA and this is F factor. But F factor is having a part of chromosomal DNA. Now when it mates with a recipient cell, then a conjugation tube is established between the two, right? And the conjugation tube, through the conjugation tube, this plasmid will break, okay? And through the conjugation tube, the plasmid will travel to the recipient cell, okay? As the plasmid travels to the recipient cell also gets the F prime factor. F prime factor means what? F factor having few genes from donor cells. Okay. So, after HFR conjugation has taken place, the F minus cell also becomes F prime cell. Okay. This F prime cell, the recipient cell is having its own chromosomal DNA plus F factor from the recipient and some genes of the donor cell. So these are the chromosomal genes of donor cell. This is F factor and this is its own DNA. Such a cell having its own DNA plus few genes from donor cell is called as partially diploid merozygote. Okay. So, F prime and F minus conjugation is virtually identical to F plus F minus mating. Once again, plasmid is transferred, but usually bacterial genes on the chromosomal DNA are not transferred. Bacterial, however, genes on F prime plasmid are transferred with it, but they are not incorporated into the recipient chromosome. Hence, the recipient cell becomes F prime and is a partially diploid merozygote or merodiploid since it has two set of genes carried by the plasmid. Okay, its own gene plus genes carried by the plasmid and hence it is a partially diploid merozygote. But recombination here also does not take place. Chances of recombination are very, very low. Chances of recombination are very high only with HFR cell. In this way, specific bacterial genes can spread rapidly throughout a bacterial population. This transfer of DNA from donor to recipient cell as a part of sex factor is called as sexduction. Now, F prime conjugation is very important to find out whether the alleles or the genes that are carried by the F prime plasmid in the merozygote are dominant genes or recessive genes. Okay. Now, F prime plasmid study is also useful in mapping the chromosome because few neighboring genes are picked up by the F factor and they are transferred. Okay. So, we can, F prime conjugation is very important for all these kind of studies to know whether the alleles are dominant or recessive for chromosomal mapping also. Okay. So, this is about conjugation. So, conjugation was a phenomena discovered by Lederberg and Tatum. Okay. And they have found out that conjugation is not similar to transformation. Conjugation is a process in which both donor and recipient in living condition, that too with in combination or in physical contact, are required. Okay. So when donor and recipient are there and a conjugation bridge is established between them, then only the transfer of genes take place. There are three types of donor: F plus cells, HFR cells and F prime cells. So that is all about